Good evening. Greetings to one and all gathered here. It gives me an warmth and great pleasure to welcome you all to the Biotalk series version 2.6. Last week, our Department of Biotechnology had successfully conducted the Biotalk series version 2.5 on the topic foodborne illness. Today, in our midst, we have an eminent person, Dr. Carolina Velasquez Pereira, who is going to speak on the topic Advancing Insights in Thrombotic Thrombocytopenic Purpura through a unique PhD track. Now, I call upon our lab HOD, Dr. Sri Kumar, to give the welcome address.
Good evening, my dear students and staff members. Hope everything is going fine and safe. I'm delighted to inaugurate this virtual BioTalk lecture series version 2.6. As a part of inauguration, I wish to highlight some of the credits of our college and the department. St. Joseph's College of Engineering, one of the top colleges in India, with the 160th position as far as the national level NIRF ranking is concerned. It has got 27 years of high reputation in the educational society with the NBA and the NAC accreditations. And it is having 10 undergraduate programs and 7 postgraduate programs with an affiliation from Anna University, Chennai. Our Department of Biotechnology was started in the year 2002 and having BTech and MTech biotechnology programs. Since inception, we have bagged 139 university ranks with 10 gold medals and 7 silver medals in it. And the department has got a sophisticated laboratory facility and fetched the research center recognition from Anna University to do PhD and research work. We have more than 1.2 crores of rupees of government funding for conducting research. Current COVID outbreak and subsequent lockdown makes us stressful by both physically and mentally. And to overcome this, our department has got the practice of conducting BioTalk lecture as a series of lectures by bringing eminent personalities through webinar and sharing their research views to the students and teachers and research personnel. Today's lecture topic is going to be Advancing Insights in TTP through a unique PhD track, which is a life-saving field. TTP, that is thrombotic thrombocytopenic purpura, is a blood disorder in which platelet clumps to form aggregates in small blood vessels, especially in the brain. In order to give more authentic research-based explanation about this TTP research, we have Dr. Carolina Velasquez Pereira, who is an assay developer at Antelope DX company from Belgium. The company actively developing home diagnostic kits to test COVID testing, sexually transmitted disease, and chronic patient monitoring kits, etc. Thank you very much, madam. We appreciate your kindness and in accepting our invitation to give a technical talk to us in spite of your busy schedule and complications due to the time zone variations. On behalf of the patrons, Dr. B. Babu Manohar, Chairman, and Madam Jesse Priya, Managing Director, and Mr. Sashi Shekhar, Director, Dr. V.S. Rao, Principal, and Dr. Parvatavardhini, Dean. I welcome the speaker, Dr. Carolina Velasquez Pereira, and all the participants to this BioTalk lecture. I also convey my warm felicitation to the Assistant Professors of Biotechnology Department, Madam Yuvarani and Madam G. Kalavati, for organizing this webinar. All the best to you all. Now I ask the program organizer, Madam Yuvarani, to give a brief introduction about the invited speaker. Over to Madam Yuvarani.
Yeah, thank you, sir. It gives me immense pleasure this evening to introduce our guest speaker of today's program. Dr. Carolina Velasquez Pereira is an assay developer at Antelope DX. And Carolina received her bachelor's degree in bacteriology and clinical laboratory in 2011 from the Industrial University of Santander, Colombia. In 2015, at the same university, she completed her master's in biomedical basic sciences. For her master's, she received the Young Researcher Fellowship by Colombian government agency called Ciencias. In 2016, she made the transfer to the KL Leuven campus, Kulak Cottage, as a PhD researcher in the laboratory for thrombosis research under guidance of Professor Karen. Dr. Carolina PhD was funded by the Horizon 2020 Marie Curie Innovative Training Center Networks. After her PhD, Dr. Pereira was a research associate in the Kim Matrinode Group Leuven Nets Lab, after which she moved to biotechnology company Antelope DX. Her expertise lies in developing diagnostic assays from the recombinant protein production to assay development and validation. Thank you, ma'am. Thank you for being with us today. And I now request you to take over the session. Thank you.
Thanks for the introduction.
Thank you.
So I will start sharing my screen, I guess. Could you please confirm if you can see my screen?
We are not seeing. Yeah, yes, ma'am. Yes, ma'am. Now we are able to see.
Okay, good. So good evening there in India, I guess at this time, and yeah, here in Belgium is afternoon. Thank you a lot for the invitation. And indeed, today I will be just giving you an overview about how was my experience as a PhD student under an European grant, and yeah, how through my PhD I was able to contribute and to develop a diagnostic assay, or like something to characterize the patients with the rare disease that is called thrombotic thrombocytopenic purpura. So in brief, it's called TTP. So the name of the talk is Advancing Insights in TTP through a Unique PhD Track. Currently, as it was mentioned, I'm an assay, the base of my PhD, I learned a lot about assay development. So that is why currently I'm working in a diagnostic company, and there I'm in charge of developing point-of-care diagnostic assays. So it's something like a pregnancy test. So I have to develop that kind of assays in that platform. So the first thing, well, here is the table of content of the talk from today. So first, I will give you a brief introduction about how I end up working on TTP, how like that took me to a research project. Like, well, there, like in a PhD, you have several projects, but here I will be presenting one part or one of the projects that I was working on. So then I will move to the aims of that project that I have, the results, and the final conclusions. So, but first, I would like to give you a brief introduction about a clinical case that it can be a real life situation. So first, let me introduce you to Daniela. She is a 23-year-old woman. And one day she started having fatigue, tiredness, headache, and like she was loose of energy. That is why she went to the hospital, and there she was diagnosed with a rare disease called thrombotic thrombocytopenic purpura. Luckily, this disease is really rare. Luckily, in that hospital, they knew how to make the diagnosis of the disease, and they make the prompt diagnosis. One of the things that the doctor mentioned was the need that she takes regular blood tests and like really follow up with medical appointments in order to avoid any relapse. But yeah, Daniela was feeling so well after she was released from the hospital that she even forgot that once she got a disease called TTP, and like she forgot the medical indications. After some time, Daniela got pregnant. And then again, she started feeling really sick this time, and so sick that she was really in an intensive unit care, unit. And that is why, she was like really sick, because it seemed that the pregnancy triggered again the disease. And unfortunately, she died. So this is a situation that can happen to someone who has this disease, thrombotic thrombocytopenic purpura. But yeah, what is thrombotic thrombocytopenic purpura? So normally, yeah, here I will give you just an overview on how the bleeding starts and stops. So we have our blood vessels, and TTP, it happens in the microvessels, so in the vascular and microcapillaries. So normally, if they got hurt, they start bleeding. And what it happens is that there is a constriction, the muscular constriction on the blood side, and the flow of the blood will decrease to slow down the bleeding. The next thing that happened, there is a protein that is called von Willebrand factor, and that is start recruiting platelets. And the platelets will go into bind together, and they will make a block, and they will seal the damaged blood vessel. And in that way, the bleeding will be stopped. But there are like several molecules involved in that case. So how von Willebrand factor is regulated, the binding of von Willebrand factor to platelet is occurring. So here I'm just showing you under normal condition how is here is the blood vessel. As you can see here in this, I'm showing the von Willebrand factor being secreted by endothelial cells. Here we see the platelets, and here the red blood cells. So what is happening normally, under normal conditions, ultra-large von Willebrand factor multimers are secreted by endothelial cells. Due to the shear stress in the flowing blood, the von Willebrand factor will unfold, and then it's going to be cleaved by a protein that is called ADAMTS13. This process, it will avoid the spontaneous interaction between von Willebrand factor and platelets. So here is just the same thing that I just told you, but in a clip. So like more like real life example.
Process, the plasma protein von Willebrand factor is important in
can you hear the sound of the video?
Yes, ma'am, we can hear, ma'am.
Okay, good. So I will start it again, just in case.
During the normal blood clotting process, the plasma protein von Willebrand factor is important in recruiting blood clotting cells, also called platelets, to damaged vessels to seal the injury and stop the bleeding. In healthy individuals, von Willebrand factor is immediately cut up into smaller pieces by an enzyme called ADAMTS13 to prevent spontaneous recruitment of platelets when blood vessels are not damaged.
So here, yeah, it was this thing that I mentioned previously. So what happens in patients like Daniela with thrombotic thrombocytopenic purpura? These patients will have a severe deficiency of ADAMTS13. So ADAMTS13 is no longer present. ADAMTS13, is being removed. That it can be, that this form of the disease has two types. So you can have an immune-mediated, that is because ADAMTS13 is being cleared by antibodies, as you can see. And the other case is the congenital TTP, and it's because there are mutations in the gene coding for ADAMTS13, and ADAMTS13 doesn't work properly. So in the absence of ADAMTS13, what happens? Ultra-large von Willebrand factors are accumulated, and the platelets will stick to it. This process is going to block arterioles and capillaries, and they will go under the circulation, they will block them, and they will produce organ failure and death when it's not treated on time. That happens to Daniela. So here is just, again, a video just saying the same things and telling which are the main organs that are affected by the disease.
Results in an accumulation of ultra-large von Willebrand factor molecules, which will bind to blood platelets and cause excessive formation of blood clots. The formation of these blood clots in small blood vessels throughout the body leads to a severe reduction in platelets, also called thrombocytopenia, a further breakdown of red blood cells referred to as hemolytic anemia, and an insufficient blood supply to tissue, also known as tissue ischemia. This results in organ dysfunction, commonly involving the brain, heart, and kidneys, and may cause acute thromboembolic events such as stroke and heart attack.
So here what I'm going to show you, so I'm going to focus mostly on the immune type of TTP. So it's when antibodies are coming and they are taking ADAMTS13 and clearing it from circulation. So here I'm showing you here is, as it was showing in the figures before, this is AMT-S13, and here what I'm doing is I'm showing you a domain structure of the protein. So it has several domains, and research on this topic has found that patients with immune mediated TTP has antibodies that are binding to different domains of AMT-S13, meaning that the immune response if TTP is polyclonal. And because the antibodies are not targeting a specific site, they are binding everywhere on the protein, and in addition is heterogeneous, and polyclonal and heterogeneous. However, most of the patients, they have antibodies that are binding to this site, the spacer domain. And some alanine scanning studies have found that there are some residues that are the main binding site of anti-spacer antibodies. This region is really critical because first, it has been shown that this region is important for the activity of of AMT-S13 on von Willebrand factor. So if there are changes on this region, or is being cleaved, the protein is going to have less activity. In addition, this protein, recent research, they have found that AMT-S13 has a closed conformation or an open conformation. And this closed conformation is mediated between the interaction of the COP domain, so it's the terminal site of the protein, with the spacer domain. So it makes a kind of clip, and that is the closed conformation of AMT-S13. So the antibodies, it seems that they are inhibiting that closed conformation. They are altering that kind of conformation. So what is missing, or why should we investigate DTP? So because as you know, or what is lacking in the field of DTP? So DTP is a really rare disease. And sometimes, as you saw from the case of Daniela, it's really important that the patients understand what is their disease, how they should take care of it. And currently there are no studies showing how much the patients understand their disease. So that is why, it's important to make studies on this topic. In addition, as I mentioned previously, it has been found that the immune response is heterogeneous and polyclonal, and it's important to understand how is the immune response, because this will help us to understand how we can develop diagnostic assays for this patient, because it's really critical that they are diagnosed on time, because if they are not diagnosed and they are given another treatment, the mortality rate is around 80%. In the other hand, it's also important to know how heterogeneous and how is the immune response, because also you can try to design therapeutics that can be you can do something there that is going to increase the activity of AMT-S13, or kind of avoid find a replacement for AMT-S13 when the normal one that is produced in the human body is not there anymore for congenital patients. So that is why it's important to investigate in this topic. And in addition, it's important to have tools that are created in the lab to advance in this field, to advance the knowledge in this field. So that is why the European Commission in 2016, they launched a program that it was the Horizon 2020, and it was a call for PhD networks. And in the lab where I did my PhD, they got that grant. So it's called Profile. So the idea was to indeed characterize the immune response in patients with DTP, and also to develop a kind of treatment for the patients. So here I'm just giving you a deep explanation on what consists Profile. So it was the name of the project. So it was a network together between the academy and the industry. So there were two academic institutions. One was KU Leuven and St. Quinn, or the University of Amsterdam. So these were the university parts of the project, like the academy. And then we have the industry, in which there were companies such as Immunodex, that is in Denmark. There was the hospital from Paris that is in France, in Paris. There were another two biotech companies, Icosagen, that is a company located in Estonia, as well as Protobios, and they are good at developing proteins. There was also PharmaTarget that is located in Holland, in Maastricht, and they are experts on in silico drug development. So that was another company that was involved in the project. And there was also BioKit that is located in Spain, and they are experts on developing of the assays. So you can see that there were several industries involved and two universities, and well, this was also supported by the European Hematology Association. So these people, they were like kind of my professors and bosses, they were part of this. And then they start hiring students from all over the world because it was a European grant. And yeah, the idea is that to have that exchange of knowledge. So they call the PhD early stage researchers. So they hire, well, Nuno is from Portugal, Kadri from Estonia, Joanna from Czech Republic, Boaz from Turkey, and me from Colombia. So we were a group of 5 PhD students from different parts of the world with different backgrounds. So in that way, yeah, it will be like a kind of training and like a constructive way of making a project. So I will now focus on my PhD track. So the idea of this kind of grant that I got, it was an industrial training network. So I was based in the University from Leuven in Belgium. So that is where I obtained my PhD at the end, and where I developed my research proposal and like I wrote the articles and so on. But then I got some kind of training and in some companies, such as first, at the beginning of my PhD, we all the PhD students, we went to the Assistance Publique Hospital from Paris, 2 hospitals, Hospital Lariboisière and Hospital Saint Antoine. And there are some experts, like they are world-leading experts on rare diseases and hematological diseases, specifically TTP. They have a reference centers for these patients. So it was really good to go there. There we saw how the patients are diagnosed and how the patients are treated, and we kind of identify what were the needs, what was missing in the field for the doctors. Like, yeah, they wanted to have a prompt test, they wanted to know how is the immune response, and in that way maybe they can modify the therapy. From the point of view of the patients, they wanted to get more informed about the disease. So all these kind of things, we learn it. We like normally you read it on a paper, that kind of things, but we saw it by first hand, what were the real needs. The next thing that I did, well, based on these ideas that we got here, just start like kind of having like, yeah, a proposal to make a project. And I was in the group of the diagnostics assays. So that is why I was sent to BioKit, that, as I mentioned, is a biotech company known because they develop diagnostic assays. And there I learned how to develop a diagnostic assay and how to make the validation. And finally, I went to Icosagen, that is the company in Estonia. And there I produced the proteins that I needed for the assay that I kind of like create here. And once I have my proteins, I went back to the university from KU Leuven, and then I started like, yeah, kind of like the assay was kind of developed, and then I started testing samples from patients that were from this hospital, amongst other places that I will talk later. And then, yeah, here I did my PhD thesis and so on. So now I'm going to start talking about the project that I work, one particular project. So the aim of this talk is going to be about the development of an ELISA using AMT-S13 AMT-S1 spacer hybrids designed previously by another researcher, but this time I expressed them in a mammalian cell line and constructed in full-length AMT-S13 to identify autoantibody hotspots in the spacer domain of AMT-S13 in a large cohort of patients. So at the end, the idea was to have an easy-to-use, inexpensive, robust, sensible, and flexible technique. And yeah, ELISA is that kind of technique. So yeah, here are a lot of terms that maybe, yeah, you were like, what is this talking about. So, well, ELISA, because as I mentioned before, is a nice and easy to use technique. AMTS13, AMTS1 spacer hybrids. So this was an idea that has been previously worked or developed by Luken et al. And he had that idea to do the epitope mapping of antibodies, so kind of like characterize the immune response of patients by making changes between AMTS13, that is our protein that is affected in patients with TTP, in changing places by regions of the spacer domain of a kind of similar protein from the same family, but yeah, in this case is AMTS1. But this protein also has a spacer domain, so that's why the regions between these two proteins could be exchanged without affecting much the expression and functionality of the protein. I mentioned here that it was made in mammalian cell lines because previously Luken did the experiments using insect cell lines. As you know, the methylation patterns can be different. So that's the post-translational modifications are different between those cell lines, and maybe that can influence the final results when you test it with humans. So that's why we express it in mammalian cell lines. And the other thing is that Luken previously, he did these hybrids in a part of the protein. So as I mentioned you, AMTS13 has several domains, and then he only took one part of those domains, a fragment. But in my case, I really wanted to express that, like make the changes and like have them expressed in full-length AMTS13. TTP is a rare disease, and yeah, like having access to a lot of patients is difficult. So the idea and the data so far has been like the biggest study has been done with 90 patients. So that is why here we really wanted to test a big cohort of patients. So the results that I obtained. So here I'm going to go in more deep about how the hybrids were constructed. So we have here the domains of AMTS, domain structure of AMTS13. So what we did is we modify the spacer domain. So the first thing is we changed completely the spacer domain of AMTS13 by the spacer domain of AMTS1. These changes allow us to know when we were doing the epitope mapping if the patient has antibodies that were targeting the spacer domain or no. Because remember that the immune response in patients with TTP is polyclonal and it binds several domains. So we really wanted to take those patients with anti-spacer antibodies. The next thing that we did is we changed fragments or regions of amino acid between 5 and 10 amino acids between the spacer domain of AMTS13 by a similar region of AMTS1. So for example, here to make the hybrid A, we changed this set of amino acids of AMTS13 by this set of amino acids of AMTS1. So that's the way that we have hybrid A. Then we have hybrid B that we change this set of amino acids by these ones of AMTS1. Then we have the hybrid C, the next set, and so on, until at the end we wanted to express 15 AMTS13, AMTS1 hybrids. So how did I do this? So I went to Icosagen, as I mentioned, and there are really experts on producing recombinant proteins. So there I was using their technology to produce this hybrid. So then I make the plasmids containing AMTS13 with the spacer domain mutated. I did the transfection, and then I have my cells that they start producing AMTS13, the hybrid with the mutation, and then I did the characterization just to know, well, it's AMTS13 secreted. So as you know, you make a Western blot and an SDS space just to be sure that there is your protein, and yeah, it's really the protein that you are searching for. So here I'm showing you the different hybrids that I wanted to create. However, remember that here I was doing modifications in the AMTS13. So that is why in some cases, these hybrids in particular, I could not produce those hybrids because probably the mutation that we were inserting, or well, the mutation, but the changes that we were making, they were affecting the secretion of the protein. So how could I know that it was affected? Because I checked the supernatant of the cells for the protein and I could not detect anything. And then what I did, it was the characterization of the of the hybrids, so meaning that I wanted to check if the hybrids were being recognized by antibodies, because what if I have an hybrid but it's not being recognized by antibody, but I don't know if it's because the patient has antibodies against this region, yes or no, or like how. So I did that kind of characterization, and I found that there was one hybrid, a hybrid F, that I checked several monoclonal antibodies against this hybrid, and I never saw binding. So it seems that this particular hybrid, the modifications that I was doing were altering the protein in a way that it was being produced, but it was not longer recognized by antibodies. So how did I do that? The fine mapping of the epitope mapping of antibodies, anti-spacer antibodies. So the first thing is that, this, of course, I learned it when I did my internship in BioKit in the diagnostic assay company. So what I did, I have my protein, but I didn't have it purified. So to coat that protein on the plate, it was difficult. So what I have is I have a monoclonal antibody that is going to capture my protein in the plate. And the next thing that I do is I add my patient antibody. If the patient antibody is able to bind to this anti-S13 with the spacer domain modified, then I add my detection antibody. So then if that binding is happening, I will see a positive signal in the ELISA, or I will see a colorimetric change. So this is a positive signal. So it means that the modifications that I'm doing here are not affecting the binding site of the antibody, meaning that that is not the epitope of the antibody, the region that I modified. But in the case that if I do the ELISA and so on, I did the modification, but the antibody of the patient is not able to bind, so I don't detect any signal at all. So if this situation happens, it means that there is epitope, because the changes that I did on the normal protein are not being recognized anymore by the antibody. So how was the methodology? So once I have my antibodies produced, so here what I'm showing you is just the crystal structure of the MDTCS domain of anti-S13. And here in this part, I'm showing the spacer domain, and the regions that I changed in the hybrids are here highlighted. So once I have my hybrids produced and so on, the next thing that I have to do, and that I did in the university, was having a large cohort of patients. As I say, TTP is rare. Having access to a lot of samples from patients is really difficult. But the lab where I did my PhD, it has a big cooperation with different reference centers in Europe dealing with the diagnostics of patients with DTP. So among them, it was the hospital from Paris and Budapest, in Marseille, and in Mainz. And there we were able to recruit a large cohort of patients. So at the end, we ended up testing 232 patients from this cohort. We have patients, we have this is the amount of patients, and from these patients, sometimes we have samples from different diseases, from different stages of the disease, meaning that there were some sample from the one patient in the acute and in the remission phase. So this is just the same thing that I previously mentioned. How was the epitope mapping done? So here is the example of a patient. So if the patient has, if the patient has antibodies, so this is just the results that I was seeing in the lab when I did the ELISA. So here I can see that this patient, okay, it has antibodies against anti-S13 because I'm seeing a signal. But if I compare when I add the anti-S13 with the spacer domain of anti-S13, what's changed by the spacer domain of anti-S1, I see a reduction. It means that this patient has antibodies binding to the spacer domain. And in which part of the spacer domain the antibodies are binding? So we can see that we still see binding for these two and these two regions, but it seems that this patient, the spacer domain antibodies are binding against the regions that change in hybrid E, G, and M. So this is the example for one patient, but this, as I mentioned before, this was a large cohort of patients. So here I'm just showing you how it looks like the epitope mapping of the patient. So here are the different regions that change of the hybrids. In red are located the regions that were not part of the epitope of anti-spacer antibodies, and in green the regions that were involved in the epitope of anti-spacer antibodies. So then based on this, you can see that here we see a kind of pattern, like are different patients, but we still see like some grouping. So we could group in this table, we could group the different profiles that we observe in the patients in the acute phase of the disease and in the remission phase of the disease. So we can see that it seems that there are some regions that are more frequent involved in the binding of anti-spacer antibodies, and we can see it here, the regions E, G, M, and there are other regions that they are not involved. However, if we compare how is the profile between the patients in the acute phase of the disease and in the remission phase, we can see that there are not so much difference. But yeah, we were able to identify profiles or kind of that fingerprint of the anti-spacer antibody binding. The next thing that we did is we wanted to check if the profile that the patient has a influence in the severity of disease. So what we did is we check the first thing, we have some clinical data from those patients. So the Benhamou score is a severity score that considers a severity score for TTP that considers the age of the patient, the LDH levels, and the cerebral involvement. So the older, the lower the LDH levels, the higher the LDH levels, and the cerebral involvement, like the worse is the patient with the disease. So we consider these parameters. In addition, we consider the treatment of the disease, the platelet count, because as it was mentioned before, the platelets are affected and normally they are decreased because they are being all binding to von Willebrand factor and they are no longer in circulation, the levels of hemoglobin and the serum creatinine. And we check all these parameters and we did not find any influence of the profile with the disease severity. And the next thing that I have to tell is like, well, as you saw from the previous graph, it seems that there are some regions in the spacer domain that are really important for antibody binding. So we call those regions hotspots. So it seems that the most of the patient has antibodies binding to these specific regions because they were like most of the samples were unable to bind when this region was exchanged with anti-S13. And here I'm just showing you in the structure where these regions are located. Then we have less immunogenic regions in the spacer domain. So they were like the ones where some patients they have, but they were not so frequently present. So those are the hybrids C, J, and N. So here in the graph, you can see where those regions are located. And there were regions that were not hotspots at all for binding of anti-spacer antibodies, meaning that when we tested, none of the patients has antibodies that were binding to this region. So where's region A, B, L, and O. So, and here in the graph, you can see where they are located. So as conclusion from this, I was able to develop a simple and high throughput screening assay for immunoprofiling of anti-spacer antibodies. We were able to screen a large cohort of patients. And we were able to do the epitope mapping of 138 patients with immune-mediated TTP. And this is the largest cohort ever studied. It was possible to stratify patients according to the anti-spacer immunoprofile. We saw that the profiles are not related with the disease severity. And most of the patient has antibodies that are binding to some regions, these amino acid regions, and they play a role in the binding of antibodies. And then the next step is to see where or what is the importance of those regions on the protein structure. So the future perspectives of this work, it was, well, one of the ideas is that will help for the designing of novel therapies, because so far as a therapy for TTP, what they want to do is they want to have an anti-S13 that is modified. And since I show there are some regions that are hotspots, so the idea is to modify those regions in a way that antibodies cannot recognize them anymore, and then the antibodies cannot bind and anti-S13 cannot be cleaved. So that can be a future therapy for these patients. So this, we are giving here information about this. The other thing is the effect of antibodies on the conformation of anti-S13. So as I mentioned before, anti-S13 can have a closed or an open conformation. So it will be interesting to see if the regions that were mutated here, they are involved in the opening or the closing of anti-S13. and identify if the antibodies that are binding to a hot, like the antibodies that are like so common or they are a hotspot for anti-spacer, the regions that are a hotspot for anti-spacer antibodies, what is the role? Are they inhibitory antibodies or they are clearance antibodies? The results from this, and if you are interested, you can find this project. It has been written in an article. So this is the article that is containing this information. And well, thank you for the invitation. Of course, I would like to thank the lab where I did my PhD. This is my PI. And here is the people from the Profile Consortium. So yeah, here PhD students and all the other people from the academy, from the industry. Here it was when I did my exchange in Icosagen, producing the proteins, and here in BioKit, developing the diagnostic assays. And of course, I would like to thank, or this work will not be possible because of, thanks to, due to the cooperation of the different universities, and of course, the patients who agree to provide the samples to make this study. Thank you.
Thank you, ma'am, for the wonderful lecture and enlightening us on the topic, advancing insights in TTP through a unique PhD track. And we have few questions in the chat box. So is that like, does these antibodies for the assay development is being prepared for the same lab or it is like outsourced?
The antibodies for the assay development, yeah, they have been previously developed, like 3H9, the antibody that was used to capture anti-S13, yes, we developed in-house. It was a monoclonal antibody, so that is why hybridoma technology developed.
Can these tests can be done in embryonic cells?
Well, I did the validation and so on for testing in plasma and serum. So then, yeah, it will be, yeah, that is a next step to check if we can test the kind of supernatant from embryonic cells or so on. But anyway, I have to, because here this test is most for antibodies. So I don't know how is the load of antibodies in the embryons. And no, anyway, no, no, it's not possible. No, you cannot test it because, yeah, you need to have antibodies to be able to use the test that I developed.
Okay, ma'am, and also like there is one more final question. Is that like in today's situation, does the vaccine, like, can it trigger the thrombocytopenia? There's a lot of questions regarding that. Like, what is your insight on that, ma'am?
Yeah, like TTP, especially, well, both cases of the both forms of the disease, it can be triggered by something. Like, it's like someone can have TTP and not knowing for a long period of time. And then all of a sudden a trigger that it is not yet so clear, but yeah, it has been seen that people with an infection can trigger the disease. So indeed, having COVID can trigger, and there are tons of articles already published mentioning that indeed TTP triggered by COVID. I just want to enlighten more about this. So I want to mention that there are other triggers. So as like, for example, the case that I show about Daniela, the relapse of TTP was because she got pregnant. So pregnancy can be also a trigger to show the symptoms of the disease. Another trigger can be some medicines, some antibiotics. They can trigger this disease. A stress situation, they have also found that can trigger this disease. So there are, yeah, those are like the main causes of developing the disease. But yeah, it's not so like, yeah, it's not so black and white that you can say, okay, I'm pregnant and you will get it. But there is a high chance. For example, in the case of Daniela, maybe she will have been under control. She will have, like, the medical doctor will have done the testing of anti-S13 to check, okay, the activity is being decreased. There are antibodies being produced. Okay, let's going to do something to avoid that you can die, or like you can have a relapse, or the fetus can be affected or something like that. So yeah, indeed, TTP can be triggered by COVID.
Thank you, ma'am, for answering the questions patiently. Now I call upon our assistant professor, Ms. Yuvarani, to give the vote of thanks.
Yeah, thank you, Alfred. On behalf of organizing team, it's my privilege to propose the vote of thanks this occasion. My heart fills with a lot of gratitude and respect for our distinguished guest, Dr. Carolina ma'am, for not only sparing your valuable time with us, but also for enlightening us with a detailed presentation on TTP through the unique PhD track. Thank you, ma'am. That was a different experience we had in listening to the development assay development process what you had given us, and also it was very nice seeing your presentation. It was well organized, and we had good animations, we had good videos, and also that was a wonderful case study you had given us with the patient samples, and we were able to understand how it is getting correlated with the process. And also you explained why it is on mammalian cells, why not on embryonic cells, stem cells, and all these works. Thank you so much, ma'am. It was interesting topic from your end. Thank you so much. Next, I would like to take this opportunity to thank our management, our beloved chairman, Dr. B. Bhavamanoharan sir, our managing director, Mrs. Jessie Priya ma'am, and director, Mr. Sasi Shekhar sir, our principal, Dr. Vaddi Seshagiri Rao sir, our dean ma'am, Dr. Parvadnavadi ma'am, for their support in conducting this event. I express my sincere thanks to our professor and head of the department, Dr. G. Srikumar sir, for his immense and continuous support in the conduction of this event. I also thank Professor Dr. Renuka ma'am for her support in this program. Next, I would like to thank my staff coordinators, Ms. G. Kalavati, assistant professor, and my student coordinator, Mr. Alfred Francis, the final year student, for their tiresome efforts in chalking out this event and making it a great success. Last but not the least, I would like to thank all my colleagues and my students for the participation and their cooperation in the smooth conduction of this event. Thanks to you all. Now, over to Alfred.
Thank you, ma'am. I once again thank all the participants for attending this webinar, and I would like to inform everyone that there will be more BioTalk upcoming series in the upcoming weeks, and also it will be communicated very shortly. Hope to see you soon. Stay safe. Thank you.
Thanks for the invitation, and good luck with the exams to all the students.
Thank you, ma'am. Thank you. Thank you for joining us today. Thank you.
Bye.
Bye.